Hi folks, welcome back. We're ready for session 4, Respond to Conditioning. I just came back from the kitchen from Thanksgiving and I could tell you everything's looking really good. The pie is out, the apple pie, the pecan pie, the turkey's going in, the stuffing is cooking, and all is well. Now along with all of these wonderful stimuli are also stimuli that are paired with the aroma of food, the taste, and the festivities. Football is on, the table is set beautifully, fine china is out. This is a great time for family and friends to be over, and I will be joining them shortly. Let's go ahead and get started with Respond to Conditioning. I'm going to go ahead and freeze the camera here a second. Okay, we're ready to start. And Respond to Conditioning starts with Pavlov. Okay, and Pavlov was known for his study and his work with the digestive system of dogs. He was awarded the Nobel Prize in Medicine for a lot of this research, but what he is most known for is this serendipity type of discovery of what he called psychic secretion. And here's how this actually happened. During his study, where he had dogs down in the basement of his laboratory, he would go down and he would place food powder into the dog's mouth, and the food powder would hit the glands of the mouth area, and Pavlov was able to actually collect secretion elicited by the dog. Now in this situation I could show you what the apparatus looked like. Right here was the apparatus part that was attached to the dog's mouth. It was surgically connected so that any saliva that comes down through the saliva ducts was actually collected into the hose over here, and by pumping this bulb right over here, this would act as a suction, and you could collect the saliva into a jar to analyze later, or you could actually collect the saliva, bring it over here, and now you could measure the output in terms of milliliters. So what you're actually looking at here, quite remarkably, very different than what Sherrington was actually looking at, is that he is using itemnotic units of measurement for studying the behavior of dogs. And now he had a way of quantifying exactly the respondent behavior that Sherrington was talking about. But rather than Sherrington talking about it in terms of observations and he was using muscle tissue, Pavlov was using a fully intact dog, using this surgical apparatus, and able to collect fine-grained analysis of output of saliva using itemnotic units of measurement. Very, very clever sort of a device to think about being invented back in the early 1900s. Let's come back to the description. So he used this device to measure the activity of dogs, in which you would place food powder to the dog's mouth, stimulus, and look at the response in terms of how much output. But what he noted was that prior to running the study, he would get everything ready in his laboratory and then walk downstairs to start preparing the dogs to run an experiment. And even before he had placed food powder into the dog's mouth, the dogs had already started engaging in the response, which is saliva. And Pavlov wondered how this could actually be. This doesn't make a whole lot of sense because he believed that food powder was the eliciting stimulus that would actually get the dog to salivate. But here he was standing before the dog without any food powder in his hand, and the dogs are already engaged in saliva eliciting, responding. He came to call this psychic secretion. And he thought that whatever he was studying was probably related to some sort of brain activity, and he decided that this phenomenon of just him walking downstairs and seeing and the dog seeing and experiencing Pavlov in their proximity, and then all of a sudden this saliva happening, was worth studying in its own right. And this really leads to something that has guided many behavior analysts over the years in the experimental analysis of behavior, and that is, when you find something interesting to study that doesn't make a lot of sense, stop what you're studying and go ahead and study this new phenomenon. In other words, it's worth studying in its own right because it should be accounted for, rather than just saying, well, this is a strange dog. and throwing the dog out of the study and maybe getting new dogs in there, Pavlov decided that it was a good idea to study this phenomenon. And using these fine-grained measures under very controlled conditions, experimental conditions, he came to talk about something called stimulus substitution. That is, Pavlov was aware that food powder would elicit responding. Somehow, some way, his presentation in the environment was a substitute stimulus for the dog salivating, although Pavlov really couldn't figure out what it was. Was it his white lab coat? Maybe. Perhaps it was his white beard, and maybe this looked like a Santa Claus. Who knows? So what Pavlov realized was that if we're going to put this under controlled conditions, he had to find a stimulus that the dog had no experience with. And that began the study of what came to be called conditioned reflex. Let's move ahead. And I'll show you the arrangement. He took a neutral stimulus, and in this case it was a bell, and he just sounded the bell. Now, if anybody owns a dog or has spent time living with a dog, you know that when you sound the bell to a dog, a lot of times they will pick their head up and look around. It is what is called an orienting response. Okay, an orienting response. And if you keep repeating this over and over and over and over again, what you're going to get is a neutral stimulus, which is a bell, which gives rise to orienting responding. Now, after a period of time, if you do this to a dog, they'll eventually stop raising their head to look at the bell. They'll stop looking at you. But each time you ring the bell, you might get the ears twitching a little bit. But what you will not get when the bell sounds is that the dog doesn't salivate, and that's the critical feature here. So no matter what the dog did to the dog did not salivate to the bell, which by definition certainly know also that when we present food to the dog, as you can see here, we have a flat line representing nothing happening in the environment. And now this box here represents that we're going to deliver food to the dog. And what happens? The dog begins to salivate. In this situation, food unconditionally will elicit salivation because that's what dogs do with the presence of food. It's an unconditional stimulus in the sense that there is nothing special that you have to do to get a dog to do this. We call food an unconditioned stimulus and abbreviate it US, unconditional stimulus. And the response to the food is unconditional. It's an unconditional response. That is, food will always happen to, salvation will always happen to food, even though the dog has no conditioning history. That's what this connection does here. You can see unconditional stimulus gives rise to unconditional response. We call this the unconditional reflex. Now, in this preparation, if we take this bell, which is neutral, and we sound the bell goes off, a little bit of time passes, and now we present food to the dog, and the dog salivates to the food, and we present this over and over again. The neutral stimulus, the bell. The unconditional stimulus, food, and this unconditional response of salivation. And we do this again and again. Now what we could do is we could do a simple test of conditioning, okay, in which we would just simply present the bell without the food, and then we would go ahead and see what happens, okay? And when we do that, we get what's called the conditional reflex. That is, we could sound the bell, and the dog will salivate. Over here, food still does what it always does, present food, and the dog will salivate. And eventually, we come down to the situation where we can present the bell alone, and the dog will salivate. Okay? And that is the conditional response. Now, I've got multiple arrangements here, and hopefully, if you have this printed out in your from your PowerPoint, you could see this a little bit clearer. I know the writing is a little bit small here. So let's go ahead and go through this. This is the variety of pairing arrangements that was used by Pavlov in his early studies. The first one is called simultaneous conditioning. With simultaneous conditioning, we have the conditional stimulus, which is the bell, and we have food, the unconditional stimulus, and we present the bell with the food within approximately 5 seconds. Sometimes it's almost overlapping. You could have the bell and the food, sometimes the bell, and then about a second or 2 seconds later the food, and so on. Okay. Now, let me pause for a moment. If you go ahead and read Pierce and Cheney, they are going to have a different description in which they will call simultaneous conditioning only when the bell and the food are paired exactly. For all sakes and purposes, for this class, and certainly for the exam, I'd rather you actually use my notes and use this arrangement and terminology here rather than using the one from Pierce and Cheney. Otherwise, it'll be a little bit confusing to you. So this is the one that you'll be tested on, okay? Bottom line. Okay, so we have simultaneous pairing or simultaneous conditioning, bell, and then within 5 seconds or less, food is presented. And you do this a number of times, and eventually you take food out, you do the test. You test to see, does now bell, which was a neutral stimulus, elicit salivation? And when it does, the neutral stimulus of a bell is now converted, and by definition, it becomes a conditional stimulus. Now, what Pavlov did was, after he did this test with one of his dogs, he got another dog from the study that had never been exposed to a bell and food and ran the next portion of the study, what he came to call trace conditioning. In this arrangement, we have the bell. It comes on, stays on for a period of time, short period of time, then it goes off, and then there is nothing happening in the environment. It is silent for maybe 15 or 20 seconds, and then food is presented. And then we repeat this arrangement again and again and again. That is, the bell comes on, stays on, goes off. Now we have a period of silence between the bell and the food, and we present food. And we do this arrangement a number of times, and we run the test again. Present the bell alone and not the food. And it turns out, when you do this arrangement with trace conditioning, the conditioning response occurs a little bit less than here in terms of the quantity, but it doesn't occur right after the bell. It occurs roughly right around here, right where food had been delivered in this arrangement. Okay, trace conditioning. Okay. Now, it turns out Pavlov used the word trace conditioning in the sense that the bell was not in the environment. Somehow the bell was there, gone, and the bell had left traces in the animal's cortex, presumably in the auditory portion of the animal's cortex. That's where he used the trace conditioning. Remember, Pavlov always tried to convert this information that he was getting here and interpret it within a neurological or physiological framework. We also see another variation on this called delay conditioning. In this arrangement, we have the bell. It comes on and now stays on for a long duration, almost the entire 20-second inter-food interval. And then as soon as the bell goes off, food is delivered. And you do this arrangement a number of times, and now you do the test of just presenting the bell goes on, stays on, and once again, the bell coming on does not give rise to immediate elicitation of salivation. It actually happens right about there. Again, very predictive that the salivation will occur roughly right before where food would have been delivered. Okay? This is called temporal conditioning, in which we don't have a salient conditional stimulus, but instead we just present food to the dog approximately every 20 seconds on an inner reinforcement or inner food interval every 20 seconds. And if you do this again and again and again, just present food to the dog every 20 seconds, then roughly what you see is every 20 seconds when you don't have food there after this arrangement, the dog begins to have little bouts of salivating every 20 seconds. Okay? Finally, we have another arrangement here we'll talk about. It's called backward conditioning. And in this arrangement, we present food first. Food's presented, goes off, and then after the food has been consumed, now we go ahead and present the bell following food. And it turns out when you do this particular arrangement, you really don't get a whole lot of conditioning, if any conditioning, which is why they call it backward conditioning. Perhaps they should be calling all of these things pairing, because what we're referring to is not necessarily the conditioning procedure, but the manner in which the bell is paired with food. Here it's paired differently than here, then here. Here nothing is paired except temporal conditioning. And here we have actually a backward arrangement. And this should give us a clue, because one of the features that makes this work is that the stimulus actually precedes the unconditional stimulus, which seems to be pretty darn important in this arrangement. And what we see here is the process of conditioning, in which it is a process in which new stimuli come to acquire the power to elicit respondents. Recall here, in all of these arrangements, food is the unconditional stimulus, and food always elicits unconditional responses of salivation. But in this case, we took a bell, which was a neutral stimulus, and we changed it so that whereas before the bell did not elicit salivation, now it does within all of these various contexts. Okay? And when that happens, we say that the new stimulus acquires the power to now elicit respondents. When we no longer present conditional stimuli, that is, if we just go back to this arrangement and again and again and again, just go ahead and present the bell in all these arrangements, but no longer pair it with food. Well, initially you'll have some conditioning happening, but if you present it again and again and again, bell, no food, and repeat this process, over a period of time, the process of extinction takes place. That is, the response, the elicited response of salivation to the bell gets weaker and weaker and weaker until it literally dies. And in fact, it follows a remarkable process. That is, when we no longer follow conditional stimuli such as the bell with food, what we end up seeing is that the magnitude of the conditional response decreases and the latency increases. Isn't that cool? Because it should be understood that in the process of conditioning, if we take that bell and pair it with food, the more salient that bell is, the louder and so forth, the more salient we make that, we get an increase in the magnitude of the response and a decrease in the latency of the response. In other words, what's really cool about this is what Sherrington had discovered that was a natural response that was in the development of the species, that when electrical stimulation hits muscle tissue and it contracts, and he developed the law of magnitude and latency from those. Pavlov, just a short time later, found that through this process of conditioning and pairing neutral stimuli with unconditional stimuli, which elicit unconditional responses, you could now have, within the lifetime of this animal, actually within moments of conditioning, that you now have stimuli that are, which were once neutral, now they acquire the properties of unconditional stimuli, in the sense that you could get an increase in the magnitude of the response and a decrease in the latency response in the conditioning process, and it reverses when we actually do the weakening here. Okay, that is, the response gets weaker and weaker and weaker, the magnitude gets weaker, and the latencies get longer and longer and longer as we expose the animal or the human to situations in which we present conditional stimuli without unconditional stimuli. That is the process of extinction. Okay. Now, interestingly enough, we find out that neutral stimuli that are very close to the conditional stimuli can also result in elicitation. In other words, if I take a particular bell and pair that bell with food delivery over and over again, not only will that bell actually elicit responses as a conditional stimulus, but other bells very, very close in sound will also have the same properties and will be able to elicit respondent behavior. A lot of these are described in Pearson Cheney. I encourage you to read the variations on this where they talk about blocking and overshadowing, although I don't believe I will be asking you that on the test. It is an interesting phenomenon that you should be familiar with in this process. Okay? All right. So let's quickly summarize here Pavlov's experiments. First of all, we find that effective conditional stimuli, such as a bell, is closely and consistently paired with unconditional stimuli. When it's closely paired, that is, bell and food within 5 seconds, within 3 seconds, or 2 seconds, we call that contiguity. That is, they're closely connected. The bell and the food are contiguous in time, particularly within 5 seconds or less. Also, that we're sounding the bell with food, bell, food, on many of the trials. If there's bell, then there's food. Okay, that refers to the contingency. And when we have situations in which neutral stimuli are very closely tied in terms of bell to food, very, very closely tied in time, and consistently happening again and again, every time there's bell and there's food, every time there's food and there's bell, when you do that, you are going to have an effective arrangement for conditioning unconditional stimuli, neutral stimuli to become conditional stimuli. This is the optimal arrangement to have here. Very close in time and consistently done, and that's one of the ways to maximize getting the conditioning effect. We also want to note here that conditioning adds new controlling stimuli, but not new responses. That is, whatever Pavlov's dog did here is nothing new that they didn't do before. In the situation before, you present food powder to the dog and they salivate. Here, through this pairing arrangement, you present bell and food, and now the bell will come to elicit the response. But the dog is still salivating. The only difference is that in the first scenario, it was food powder that gave rise to eliciting responding. In the second scenario, after conditioning, now a bell gives rise to salivating. But the dog is really not doing anything remarkably new. In other words, when we talk about the things that humans do that are very, very different, a lot of that involves a different type of learning known as operant conditioning and operant learning. So this is a very different kind of learning process. Third, we note here that, as I mentioned before, continuous pairing is optimal for conditioning to take place, for conditional stimuli to acquire power, but it's not absolutely required for conditioning to occur. That is, this would be ideal conditions, but there are phenomena that actually happen, what we might call one-trial learning. This might actually happen when we develop taste aversions. That is, you may be able to eat certain foods, but one time, and on one particular occasion, you were eating maybe raw oysters. And in the process of eating raw oysters, you also may have eaten something which was bad in the raw oyster, or you might have actually been drinking a little bit too much alcohol, and you get deathly sick from your one and only time that you ate raw oysters. And you were so sick that anytime you come near raw oysters in the future, you already start feeling queasy, even though you're not even eating it. And it's interesting, the sight of it, the smell of it, and so on. To me, what's interesting about taste aversions is that we usually have an aversion to food that has a very unique smell, taste, or texture. Now, usually when I teach my class, it's an opportunity for me to ask students to go around the room and raise their hand and describe any taste aversion that they have. And it's funny that when I do this, I almost always have at least 2/3 of the class that will say there are certain foods they can't eat. One guy, I remember, said, I cannot eat mushrooms. They drive me nuts. Another person would say seafood. Seafood, by the way, is a very popular taste aversion thing because of the fact that it's got that unique texture. The same thing with eating raw eggs. People don't like that. Eating foods that have particular kinds of smells, like if a person eats Indian food and which has a lot of curry, anything that has curry will get them sick. Certain kinds of Chinese food which have distinct flavors and smells will actually get them sick. With me, I have to tell you, number one is tequila for obvious reasons. But I cannot go near the stuff. I don't even like looking at it, smelling it. I can't eat I drink margaritas, salted or unsalted. I really can't do it. I have that one trial learning that makes it so that I have to drink usually 7-Up when I go eat at Mexican restaurants. Oh, woe is me. Okay, let's move on. One of the other things that is remarkable about Pavlov's work is that in all of his explanations, he did not really appeal to explanatory fictions. He always believed it to actually be something had to be going on neurologically to explain this. But he really didn't use kinds of explanations that we might use to explain it. For example, we might say that through Pavlov's work, Pavlov's dog believed that food was going to follow the bell. The dog had the intention that food was going to follow the bell, or the dog expected food to be followed by the bell. In all of these situations, we are making appeal to something that we never really saw. We don't see these things such as beliefs, intentions, and expectations. Okay? And in fact, we might even go on to say, that Maybe the dog associated the bell with the food. That might be an appeal. An association kind of connection was made between bell and food, and the dog made this. When in fact, we know from our end that in fact the dog did not make any association. It was in fact Pavlov who associated bell with food. But we're going to come back and talk about this later on, that when we see things that happen, there is an immediate appeal for us to start talking about the results of experiments and putting the causes inside the body of the organism. In this case, that's what we're doing with Pavlov's dog. When we say the dog is doing the associating, we are kind of misleading the facts as we see them. Pavlov did all the arrangements, and you saw all the various arrangements of delay conditioning, temporal conditioning, trace conditioning. That wasn't the dog doing that. That was Pavlov doing that. And we note here that we come back to what I mentioned on the very first session about not having an explanation and not making up explanations when we are going beyond our facts. The same thing here is true. A satisfactory account of the conditions of secretion using objective facts in the recent history of the organism, in this case, the dog. That's all we really need to know. If we want to know under what conditions will Pavlov's dog salivate, here they are. We could lay them out. We could predict and control these arrangements under controlled conditions. And we know these things to be pretty reliable in the sense that this has been replicated many, many times in the science of behavior. And we've gotten pretty close reliability every single time across many different species. That it doesn't matter the species we're talking about. They all seem to give you the same generalized results. Okay. And we are going to stop here. After Pavlov's work, we're going to take a break, and we will come back to the next session, which would be session 5, and we will talk more about Watson's work and some of the work of respondent conditioning and respondent extinction as we use this to get rid of people's fears and phobias using techniques such as systematic desensitization and implosion therapy and so on. In the meantime, things are starting to smell really good